Welcome to this biology investigation planning video. Today, we'll learn how to plan an experiment to investigate the effect of temperature on osmosis in plant tissue. This is a common biology practical that tests your understanding of experimental design and osmosis concepts. Let's start by understanding what osmosis is. Osmosis is the movement of water molecules from an area of lower solute concentration to an area of higher solute concentration through a partially permeable membrane. In plant cells, the cell membrane acts as this partially permeable membrane, allowing water to move in or out of the cell depending on the concentration gradient. The aim of our investigation is to determine how temperature affects the rate of osmosis in plant tissue. Temperature affects the kinetic energy of molecules, so we expect it to influence how quickly water moves through cell membranes. For our experiment, we'll use potato tissue, which is commonly used in osmosis investigations because it's easy to obtain, cut into uniform pieces, and measure changes in. When designing an investigation, we need to clearly identify our variables. The independent variable is what we deliberately change. In this case, it's the temperature of the water baths. We'll use 5 different temperatures: 5 degrees Celsius, 15 degrees, 25 degrees room temperature, 35 degrees, and 45 degrees Celsius. The dependent variable is what we measure to see the effect of our changes. We'll measure the percentage change in mass of the potato cylinders after they've been immersed in distilled water for a set time. This change in mass indicates how much water has moved into or out of the potato tissue by osmosis. We also need to control all other variables that might affect our results: the type and source of plant tissue. We'll use cylinders from the same potato, the size and initial mass of the potato cylinders, the concentration of the external solution. We'll use distilled water, the volume of solution in each beaker, the duration of immersion, the method of drying the potato cylinders before weighing. Now, let's outline our experimental method. We'll need the following materials: a large potato, a cork borer to cut uniform cylinders, a scalpel and ruler to trim them to equal lengths, an electronic balance accurate to at least 0.01 grams, beakers for the water samples, 5 water baths at different temperatures, thermometers to monitor the temperatures, distilled water, paper towels for drying the cylinders, a stopwatch to time the experiment. Here's our step-by-step method. First, we'll prepare our potato cylinders. We'll cut at least 15 cylinders from the same potato using the cork borer. Then we'll trim all cylinders to exactly 3 centimeters in length using the scalpel and ruler. This ensures a consistent surface area to volume ratio, which is important for osmosis. Next, we'll set up our 5 water baths at the different temperatures: 5, 15, 25, 35, and 45 degrees Celsius. We'll pour 50 milliliters of distilled water into 15 beakers, 3 for each temperature, and place them in the appropriate water baths to equilibrate. For each cylinder, we'll gently blot it dry with paper towel, measure and record its initial mass, place it in a beaker of distilled water at the assigned temperature. We'll have 3 cylinders for each temperature as repeats. After 30 minutes, we'll remove each cylinder from its beaker, gently blot it dry using a consistent method, immediately measure and record its final mass. We'll then calculate the percentage change in mass for each cylinder and find the mean for each temperature treatment. After collecting our data, we'll need to analyze it. First, we'll plot a graph with temperature on the x-axis and mean percentage change in mass on the y-axis. We'll draw a line of best fit to show the trend. If our hypothesis is correct, we should see that higher temperatures lead to greater water uptake by osmosis, shown by a higher percentage increase in mass. This happens because higher temperatures increase the kinetic energy of water molecules, making them move more quickly through the partially permeable membrane of the potato cells. Finally, let's evaluate our investigation method. Strengths of this method include: we used multiple repeats at each temperature to increase reliability. We controlled key variables like potato source, cylinder size, and immersion time. We used a wide range of temperatures to clearly see the trend. Limitations might include difficulty maintaining exact temperatures throughout the experiment, variations in the cellular structure within the potato, potential inconsistency in drying the cylinders. We could improve the investigation by using more repeats to increase statistical validity, using a data logger to continuously monitor temperatures, measuring more time points to see the rate of osmosis over time. In conclusion, this investigation should give us clear evidence of how temperature affects osmosis in plant tissue. The combination of controls, repeats, and appropriate measurements ensures we collect valid and reliable data to test our hypothesis. To summarize our plan, we'll investigate how temperature affects osmosis in potato tissue by measuring the change in mass of potato cylinders after immersion in distilled water at 5 different temperatures. We've designed a procedure that controls important variables, includes repeats for reliability, and will allow us to clearly see the relationship between temperature and the rate of osmosis. Remember that careful planning is essential for any successful scientific investigation. Thank you for watching.